today's lecture is about A-Levels Unit 2 and Topic 1, and this lecture will be divided into parts, and this is Lecture 1. Today's topic is observation of cell. So what is a cell? Cell is a microscopic structure, right? And we need and to see the cell we need magnifying tools like a microscope to see what is within a cell. Okay, and our overall topic is about all these things. So let's get started. Observing cell. Cell is the microscopic structure, and we need magnifying instruments like microscope to get idea about all cell organelles. And before knowing this function of cell organelles, their structure should be known, right? Like before knowing the function of all the cells, cell organelles, their structure should be known. For example, the structure of chloroplast or mitochondria, their structure should be known before we know the function of them, right? It's just like that. So our next part is about magnification and resolution. Okay, magnification and resolution. On the basis of resolution and magnification, microscope is classified into two categories. They are light microscope and electron microscope. Okay, two types of microscope. One is light and one is electron, and they are differentiated by the magnification and resolution of it. Okay, let's know more about magnification and resolution. In light microscope, beam of light is used, whereas in electron microscope, beam of electron is used. Like in light microscope, we can see through the cell by the beam of light which is used in it, and in case of electron microscope, electron is used. Okay, and magnification is the capability. Like what is magnification and what is resolution? More specifically, magnification is the capability to compare two objects. It can also be defined as how many times an object is bigger than the real one. It is expressed as 2 times. And whereas resolution is the capability to discrete 2 points of an image, or resolution is a measure of how apart 2 objects must be before we see them separately. Okay, 0.1 mm, whereas the resolution of electron microscope is 0.2 micrometer, right? It's huge. Like we can see like a more in it and more clear picture. Now let's go to what is light microscope and electron microscope on that topic. Light microscope and electron microscope. A microscope was first used in the UK, like the background history, but this one was not efficient. It could only magnify into for 20 times. Robert Hooke used a magnifying glass to observe some metals and fabrics. Later on, magnification capabilities enriched by other scientists. Okay, nowadays light microscope can magnify an object up to 1500, while an electron microscope can magnify up to 5 times. In light microscope, a beam of light is allowed to pass through the object. As I said before, in light microscope, light is used to see through the cells and objects. Okay, it is allowed to magnify using 2 consecutive lenses, objective lens and eyepiece lens. like how many lenses are used for using a light microscope. It uses two consecutive lenses, objective lens and an eyepiece lens. All right. Now we will do a little comparison between light and electron microscope. Okay, light microscope and electron microscope. I hope it's clear to see. Alright, okay. Light microscope. It has limited magnification power, i.e. for 1500 for maximum. Alright. And for electron microscope, it has huge magnification capability, as I said before, for up to 5 lakh maximum. Limited resolution. A microscope has limited resolution, while the electron microscope has huge resolution. Can be used to observe both living and non-living image. Like it is an advantage for using a light microscope that we can observe both living and non-living cells, whereas in case of electron microscope, during the preparation of samples, objects get dead, so no living image is found. Like we cannot see the living process in it, we just can have the dead image of an organism or of a cell. Okay. Both colored and normal image can be formed in light microscope. Another advantage for light microscope that we can have a color image and both normal and color image, but in case of electron microscope, only black and white image is formed, no color image. Artifact for our form, it's a disadvantage for light microscope because it's not purely original. Okay, it has some artifact, and less chance of to form artifact is an advantage for electron microscope, like more realistic image. It's cheap and available. Like it's also expensive, but in comparison to electron microscope, it's quite cheap and available, and of course it's expensive. Handling of light microscope is comparatively easier, and it's the electron microscope is quite difficult to handle. All right, so this is the end of our video. I'll be giving you an extra question just to see that, just for you to practice it at home. All right, I'll be giving its answer in the next video for Unit 2. What is the difference between magnification and resolution? Okay. Thank you so much for watching my video. Thank you.